So this is an extremely exciting field of research in multiple myeloma. And I'm very excited to be presenting the latest data at the Friday Scientific Workshop on multiple myeloma. So I have been working for the past 10 years on genomics applied to evolution of multiple myeloma. And that has discovered how after diagnosis, the disease is very heterogeneous and very complex and may evolve in multiple ways owing to increased chemoresistance. And we know that multiple myeloma is not curable, probably because disease can easily evolve. Now, what we have found in the latest years was that the application of these genomic techniques to asymptomatic and preclinical stages has highlighted how the asymptomatic stages, namely MGUS and smoldering multiple myeloma, are quite heterogeneous themselves and may actually reveal how these people affected by these conditions may progress or not progress, and that is a major clinical question. So in particular, I think the major progress resides in our ability to use genomics to predict how asymptomatic conditions will behave in the coming years of the patient. Our studies, together with studies from the Dana-Farber group, from Irene Gabriel, have highlighted a number of genomic features of asymptomatic conditions that may predict future progression. And that's way beyond our abilities in the clinic, our current abilities in the clinic. These abnormalities pertain to complex rearrangements, to specific gene mutations, to the activity of particular mutational processes. And there is a crescendo, there is a climax of these events from the very early and indolent conditions that may never progress for life to the ones that actually are more advanced and at high risk of progression. So in this scientific workshop, I will review all these latest findings and also highlight how the genomics of asymptomatic myeloma conditions may in the future reshape our classification of people with monoclonal gammopathies and how we deal with them in terms of follow-up and therapeutic decisions.